Introduction to Histology, Cytology and Embryology. Main questions. Histology, this word derived from the Greek word histos, tissue, and logos, science. It's science about structure, development, and functions of the body's tissues. It studies mainly the microscopic structure of the body. Our course consists of 4 parts: general histology, science about the tissues; particular histology, study of the microscopic structure of organs, microscopic anatomy; cytology, or cell biology; and embryology, science about embryo development. That was the full name of the present course, and our department is histology, cytology, and embryology. We will start this cell biology, then we will study general histology, epithelial, connective tissue, muscle tissues, and nervous tissues. Then move to particular histology. We will study the structure of all organs of the body. And finally, you will study the embryology to understand the source of development of tissues and organs in embryogenesis. The place of histology in the system of medical knowledge. Histology is the basic fundamental science. It lay in the basis of your medical knowledge. So it's extremely necessary to understand the further medical science like biochemistry, physiology, pathologic anatomy, and pathologic physiology, and then surgery, therapeutics, and so on. As compared to anatomy, histology studies the microscopic structure of the body, the microscopic structure of any organ. It means tissue, cellular and subcellular organization of organs. And main research methods in histology are microscopic methods. This is a picture of modern research light microscope. To study the structure in microscope, it's necessary to prepare the histological preparation first. Prepare the thin section of organ, and specially stain it. The types of dyes and staining used in histology. Type of dyes: basic, like hematoxylin, and ability to be stained by basic dyes called basophilia, allowing the basic dyes. Acid dyes like eosin, and ability to be stained by acid dyes called oxyphilia. Polychromatophilia, ability to stain by both types of the dyes, both basic and acid. And metachromasia, ability of structure to be stained in the color different from the color of the dye in a solution. For example, the blue dye stains the structures in red. These microphotograms demonstrate the staining by basic hematoxylin and acid azine dyes. And this is a double hematoxylin and azine staining. And you see that basic dyes stain specifically the nuclei of cells, which contain a lot of acid, DNA. Sometimes the basic dyes stain cytoplasm also, because it contains RNA, especially in ribosomes of cytoplasm. Acid dye stain mostly basic proteins of cytoplasm. When you use double staining, first hematoxylin, then azine staining of sections, you have got very nice staining both of nuclei and cytoplasm. This is an example of fluorescent microscopy. You see the knife cells stained by fluorescent dye. Histochemistry is detection of enzyme activities, of the content of substances like lipid, carbohydrates, proteins, nucleic acids in the sections of tissue and organs. So sometimes histochemistry called topographic biochemistry. Immunohistochemistry can indicate the location of antigen by marked antibodies. This is a section of organ and antigen location, and if you prepare antibodies against those antigens and marked them by fluorescent dye under light microscope, and this fluorescence will indicate the location of antigens in tissues and organs. You can use the indirect methods when you prepare the secondary antibodies against the primary antibodies and marked them by fluorescent dye. Electron microscopy uses the electron beam instead of visual light. There are two types of electron microscopy: transmission and scanning. This is a picture of transmission electron microscope, and you can get the magnification up to 1,000,000 times and very high resolution, less than nanometer. This is transmission electron micrograph of the blood capillary transverse section. You see the lumen, this blood, former elements like erythrocyte and platelet. You see the wall, capillary wall, consists of endothelial cells. Scanning electron microscopy, you can get the image in 3D. For example, you see the picture, then spermatozoa attack ovum during fertilization. Freezing fracturing methods, followed by scanning electron microscopy. This is a picture of the cell. You see the nucleus and organelles, and here you see the kidney, renal body, and without glomerulus, and surrounding renal tubules. In this method, you freeze deeply the cells or organ and crush it with special knife, then cover by metal ions and study in scanning electron microscopy. After radiography, used the substances like glucose or amino acid marked by isotope. Microphotography in computer image analysis. In modern microscopes, use the digital video cameras which accept the image you get through the microscope and convey it to the computer, and this image appears at the screen of monitor. And using the special computer program, you can treat these images. You can measure the size, the shape of structures. You can also measure the optic density of the stain structure and make a conclusion about activity of enzymes or contents of some substances in tissue and cells. So the research methods of histology, microscopic methods, and main types of microscopes are light microscope, transmission, and scanning electron microscope. In light microscope, you use a source of visual light which condense it in condenser by lens and enter through the spaceman histological preparation, and the image increased by objective lens and then increased by ocular lens, and the image enter into your eye, and you can see the increased image of spaceman. In electron microscope, use the beam of electron which leave the cathode when between cathode and anode the about 100,000 volt appeared, and the beam of electrons go through the condenser lens. It's electromagnetic field. We concentrate the beam of electron into the spaceman, then it increased by magnetic field, objective, like a objective lens, and magnification increased by another magnetic field as a projection lens, and increased image of the spaceman appeared at the fluorescence viewing screen, and you can see it through the viewing window. In scanning electron microscope, for a narrow beam of electron, scanning the surface of spaceman, and electron reflects from the surface and accepted by electron detector and amplified, and you can observe the image of spaceman in the television monitor in 3D. It's no problems to see the objects in 3D in scanning electron microscope, but it's difficult problem when you use usual light microscope or transmission electron microscope, because you can see the two-dimension image of three-dimension structures which really exist in your mind. Convert the two-dimension image into a real three-dimension image of structures. Units of length used in histology for the measurements of cells. In light microscopy used micrometer, 10⁻⁶ degree of meter. In the electron microscopy for the measurements of cells ultrastructure, the nanometer used, it's 10⁻⁹ degree of meter. As an example, the size of erythrocyte is about 8 microns, and size of ribosomes 25 nanometers. History of histology. There are three main periods in the development of histology as a science. The long period before microscopes development. The outstanding scientists, ancient scientists like Aristotle, Galen, Avicenna, Vesalius, try to suggest the structure of the body, human body, and its organs and tissues without microscope, just by naked eye. And then microscopic period started about 3.5 years ago, when the first microscopes were developed. The first microscopes were very primitive, but they provide the magnification up to 300–400, and scientists can see the small structures invisible without microscope. And the first inventors of microscope were Galileo Galilei, the father and son Janssens, and Cornelius Drebbel, Robert Hooke, and then microscope became more and more precise, appeared achromatic microscopes, and the more and more data about microscopic structure of the various animals and plants and human body, about microscopic structure of different organs and tissue, accumulated. 1838, the Schwann and Schleiden formulated the cell theory. And in the middle of the 19th century, the special science histology appeared in the world, and the first school of histologists appeared in Russia, initially in Moscow, St. Petersburg, and Kazan universities. And third modern period of histology development started in the middle of 20th century. Then the methods of electron microscopy, histochemistry, and immunohistochemistry, as well as computer image analysis, appeared. So, in definition of cell, the smallest alive part of the human body, consisting of cytoplasm and nucleus, which provides the basis of structure, development, and functions of the body, and depends on its regulatory mechanisms. Cells possesses all 5 things of an alive structure: definite structural organization, metabolism, exchange of substances with the environment, constant self-renovation and reproduction, irritability and excitability, and movement. Cell theory. First, cell is the smallest part of the alive organism. Cells of the different organisms have a similar structure. Every cell is derived from another cell, every cell from cell only. Organisms are the complex ensembles of cells and their derivatives united into the system of tissues and organs. There are a huge amount of cells in the body, and the typical cell of the human body consists of the nucleus and cytoplasm. It's separated from the environment by cell membrane. There are more than 200 types of cells in the body. All of them have the above-mentioned plan of structure, but they considerably varied in shape. You can find the columnar, cuboidal, flat cells, round, the cells with long processes, the spindle-like cells, goblet cells, these flagella, and so on. The size of cells varied between 5 and 150 microns. In addition, there are derivatives of cells. They are postcellular structures like erythrocytes or platelets, supracellular structures like symplasts containing a lot of nuclei and huge amount of cytoplasm, and intercellular matrix. The example of symplast is skeletal muscle fiber. You can see these fibers under your microscopes during the class. You can see a huge amount of cytoplasm and many nuclei at the periphery of symplasts. You can see also a loose connective tissue which consists of cells. At low magnification they looks like points, and at high magnification you can see their different shape, and their derivatives intercellular matrix, which consist of ground substance and fibers, collagen, thick bundles of collagen fibers, and separate thin, dark, branching, elastic fibers.